Hello, everybody. It's 11 o'clock Eastern Standard Time, and I want to welcome Koen van de Wetering. He is a professor and our PXE researcher at Thomas Jefferson University. And the name of this talk is ABCC6, Regulation of Extracellular Pyrophosphate and PXE. And I will let Koen take it over from starting now. Thank you.
Okay. Thank you, Mary, for your
Welcome.
introduction. Today I will talk about ABCC6, extracellular pyrophosphate, what the extracellular pyrophosphate does, and the relation with PXE. And before starting my presentation, I want to thank upfront PXE International for their long-term support of our work. And if you have any questions, please don't hesitate. I wouldn't mind if you ask anything during the presentation. I don't know how it's normally done, if that's okay. Is that okay? Yeah, okay. Or at the end, it doesn't matter, actually. Okay, first something a little bit about PXE and the relation with ABCC6. So Pseudoxanthoma elasticum, or PXE, as most of you will know, is most often caused by absence of ABCC6, so by mutations in the gene encoding ABCC6. There are a few exceptions, but let's say in 95% of cases, ABCC6 is affected in PXEers. It's a late onset disease, which means that the first signs appear around puberty, so in the second decade of life. And it's a prototype of a connective tissue disorder, which means that it affects soft tissues, especially blood vessels, eyes, and skin. And the clinical manifestations that are normally encountered in PXEs are yellowish papules, inelastic skin, vision loss, that's an important one, I think, and intermittent claudication, which is caused by narrowing of arteries in the legs. So on this slide, I put a couple of clinical manifestations that are probably very familiar to you. So PXE most often starts with yellowish papules in the neck region, which then can coalesce and form larger areas of inelastic material. And that results in the skin losing its elasticity and getting a leathery or wrinkled appearance. So when it comes to the blood vessel involvement, you can see that arteries are affected, and that can be seen in this CT scan. CT is now sometimes used to follow disease progression, also in PXEs. And you can see that here, in this case, an artery is calcified. And I will come back what calcification is in the next couple of slides. And this calcification results in narrowing of the lumen of the arteries, and that will in turn affect the normal functioning of these blood vessels. For instance, sometimes not enough blood flow is going to the muscles in the leg. And what PXE patients, at least that's what I've always been told, so correct me if I'm wrong, what PXE patients fear most is actually what's happening in their eyes. Because due to the calcification, which also happens in the eyes, PXE patients tend to lose visual acuity over time, at least if the disease goes untreated. So if we focus on what's actually happening at the sites that are affected by the disease, then you will see that affected regions show ectopic calcification. And what is ectopic calcification? Ectopic calcification is the precipitation of calcium and phosphate. And calcium and phosphate often precipitate around elastic fibers in PXE patients. And that's why skin can be affected. So this is an histological staining of a biopsy taken from an affected region of the skin of a PXE patient. And this is the outside of the skin, that's called the epidermis. And this is the more down part of the skin, so the dermal layer of the skin. And in the dermal layer of the skin, there you see the black material, which is calcium phosphate. And that's hard material. It's the same material that's present in your bone and bones. And calcium phosphate in the skin damages the elastic structures that are normally present. And that explains why the skin gets a wrinkled and sometimes leathery appearance. So the same thing, calcification, also happens in blood vessels, in arteries. And an example is shown here. This is not an artery of a PXEer, but this is an artery of a PXE mouse, because we have a mouse model for human PXE. And these animals have very similar clinical manifestations as human PXEers. They also have calcification in their skin. They suffer from calcification in their eyes and also calcification in their blood vessels. So calcium phosphate precipitation is what is causing the problems in PXEers. And what is calcium phosphate precipitation? Well, calcium and phosphate are two ions. Calcium has two positive charges, and phosphate, or inorganic phosphate, as it's also known, or Pi, has negative charges. And that depends a little bit on the form of the ion, but it can have 3 negative charges, 2 or 1. And therefore, because they have opposite charges, they tend to combine and precipitate. And that's known as mineralization or calcification. So if they get together, they precipitate. And the concentrations of calcium and phosphate everywhere in the human body are relatively high. So everywhere in the body, calcium and phosphate tend to combine and precipitate. And this process is not always bad. Let me stress that, because that process underlies normal bone and tooth formation. So you need it. But sometimes calcium and phosphate also precipitate in soft tissues. And that's known as ectopic calcification. And the same mineral as found in your bones and teeth is found then in the soft tissues. This should not happen, obviously. So several mechanisms are present in your body that prevent the precipitation of calcium and phosphate in soft tissues. And one of the molecules that's crucial to prevent the precipitation of calcium and phosphate in soft tissues is pyrophosphate, or inorganic pyrophosphate, or PPI. And PXE patients, or people with PXE, I should say, have low levels of this mineralization inhibitor pyrophosphate in their blood. And that explains why sometimes in their skin, in their eyes, and in their blood vessels, calcium and phosphate combine and precipitate and give problems. So this is a rather a chemical slide, because here I depicted the chemical structure of inorganic phosphate and inorganic pyrophosphate. And the only reason that I'm doing this is to show you that these molecules are very similar. So they don't differ that much. Because in fact, inorganic pyrophosphate, so the molecule that inhibits the calcification, consists of two molecules of inorganic phosphate. They're combined to form pyrophosphate. And it's also the case that in your body, inorganic pyrophosphate can be converted into inorganic phosphate. There is an enzyme, and you can see an enzyme as, let's say, a big scissors that cleaves this bond, that cleaves pyrophosphate to form two molecules of phosphate, which in theory can stimulate calcification because it's part of calcium phosphate, for instance, in the bone. Okay, then something about the relation between PXE, ABCC6, and pyrophosphate. So how does ABCC6 regulate levels of pyrophosphate, and how is that process altered in PXEers? So as said, mutations in ABCC6 underlie PXE. And ABCC6, so the protein that's missing in PXEers, is in unaffected people only present in the liver. So ABCC6 is not found in the eyes, it's not found in the blood vessels, and it's not found in the skin. And that means that PXE is a metabolic disease that's due to the absence of pyrophosphate in the circulation, and ABCC6 is normally providing pyrophosphate to the periphery from the cells of the liver. How does this work? Because it's quite a complicated process that results in pyrophosphate regulation by ABCC6. Because ABCC6 is a large protein and it's present in the plasma membrane of the liver cells. And the plasma membrane separates the interior from the exterior. And what ABCC6 does in this plasma membrane is it functions as an efflux pump. So it pumps something from this is a cell, and it pumps something from the inside of the cell into the extracellular environment, into the blood, basically. There are many ABC transporters, and depending on the specific ABC transporter, the substrates that they pump into the extracellular environment are, for instance, hormone, metabolic waste products, cyclic nucleotides, bile acids, etc. But ABCC6 does something different. So what ABCC6 does, it's not directly pumping out pyrophosphate. Instead, it pumps out a precursor of pyrophosphate that's known as ATP. So from the liver cells, ABCC6 pumps ATP into the blood, and then in the blood, the ATP is converted into pyrophosphate. And next to that, also another molecule is formed out of the ATP, AMP. But for now, we will not focus on AMP. We will mainly focus on pyrophosphate because that's the crucial metabolite that prevents calcium and phosphate to precipitate. And what we found basically is that in PXE mice, which are also known as ABCC6 minus mice, the levels of AMP and pyrophosphate in the blood are different. So ABCC6 minus mice, so the PXE mice, have reduced levels of pyrophosphate going out of their liver. So they produce less pyrophosphate in the blood circulation. And in real human PXEers, this results in lower levels of pyrophosphate in the blood compared to people that are not affected by PXE. So this is approximately 30% of the levels found in people that are not affected by PXE. So in a cartoon, I here show how ABCC6 prevents ectopic calcification. So in the liver cells, ABCC6 pumps ATP into the blood, and then you need another enzyme to convert the ATP into pyrophosphate, the calcification inhibitor. And the pyrophosphate then travels to the eyes, to the skin, and there prevents the precipitation of calcium and phosphate. So we discovered this already a couple of years ago. And after after we had reported that the central problem in PXEers was low levels of pyrophosphate, then several other groups also started to work in trying to find ways to increase the level of pyrophosphate in the blood. Because in theory, if you would be able to increase pyrophosphate in the blood, you should be able to stop the disease from progressing. And you should then start treating as soon as possible. And my initial schematic overview, how ABCC6 regulates extracellular levels of pyrophosphate, is a little bit oversimplified because there are other proteins that also contribute to the regulation of pyrophosphate in the blood. And one very important protein in this respect is tissue non-specific alkaline phosphatase. So whereas ABCC6 and EMP-P1 are involved in generation of pyrophosphate, so they produce pyrophosphate, tissue non-specific alkaline phosphatase, this enzyme, TNAP, is involved in the degradation of pyrophosphate. So it keeps levels low. And there's now a pharmaceutical company that's focusing on the development of an inhibitor of this protein. Because if you would inhibit the degradation of pyrophosphate, then you could increase the levels in PXEers to levels that are sufficient to prevent calcium phosphate precipitation. There's also a company that's trying something else. This company focuses on the enzyme that forms pyrophosphate out of the ATP released into the extracellular environment. They propose that if you give more of this enzyme, that ATP that's released into the blood via mechanisms that do not depend on ABCC6, and I will come back to that later in my webinar, then you could also increase levels of pyrophosphate in the blood, and that should again stop the disease from progressing. And another approach that has been tried in Utrecht, so that's where I went to vet school because I'm a vet by training. In Utrecht, that's the Netherlands where I'm from, there they tried something else because ethidronate, which is a bisphosphonate, is a molecule that looks very much like pyrophosphate. It's a drug that can be given to people that suffer from ectopic calcification. And in Utrecht, they have shown that if you give ethidronate, that slows down the disease from progressing. So there's some success in using ethidronate to treat PXE. Okay, but the focus of my today's webinar is slightly different because we focused on something else. The first part will focus on testing the hypothesis that it's possible to stimulate release of ATP from cells that is independent of ABCC6. And then this ATP can be converted into pyrophosphate in the blood because cells can use other transporters than ABCC6 to release ATP in the blood. That's known. It was, however, not known which transporters are involved. And we thought that if we know which transporters are involved, maybe we can look for drugs that can stimulate these alternative transporters. And in my next slides, I will come back and explain this idea in more detail. Another way to treat PXE I will focus on is the oral administration of pyrophosphate. You will probably have heard several times people talking about, is it possible to give pyrophosphate as a pill? And it has been shown that there's some uptake in mice and humans, but as I showed you earlier in this webinar, that might be a double-edged sword. That was at least my hypothesis, Because if you give a lot of pyrophosphate, it can give rise to increases in organic phosphate. So, and that can increase calcification. At least that's what we hypothesized. So we worked on that also a little bit. But let me first start with our work on trying to identify proteins that do something similar as ABCC6. And why did we think this is a good way to try to find new avenues to treat this disease? Well, PXE patients, they still have some pyrophosphate in their blood. It's not completely absent. And I won't go into detail, but also this amount of pyrophosphate comes from ATP that is released by cells into the blood. And in another slide, in another cartoon, that can be depicted as follows. So in healthy individuals, so people that do not have PXE, most of the pyrophosphate that's found in the blood comes from ATP that's being released by ABCC6 into the blood and then converted by EMPP1 into pyrophosphate and AMP. There are, however, also other transporters doing a similar thing. So they also pump out ATP, and then again, EMPP1 converts the ATP into pyrophosphate. So PXEers miss ABCC6, so they miss the ABCC6 dependent hepatic ATP release. So they miss a large part of the pyrophosphate that's normally present in the blood, but they still have these other ATP release pathways. So we thought that if we know which other transporters do the same, maybe we can stimulate these transporters so they start to release more ATP, and then more pyrophosphate can be formed in the blood of PXEers. And that should again slow the disease from progressing, or maybe completely halt the disease from progressing. Okay, so there are several ways that cells can use to get ATP into the blood circulation. And the two most well-known processes are regulated exocytosis. And what's that? That's there are structures inside cells that can be loaded with ATP. And you can see these structures as, let's say, balloons filled with ATP, and these balloons travel to the plasma membrane and they're then emptied into the extracellular environment. So that's one way that cells can use to get ATP into the extracellular environment. And next to this regulated process of exocytosis, there are also other transporters that are known to be involved in getting ATP into the extracellular environment. And here are the names of a few of them. We had, however, a different hypothesis because we focused on a protein that's known as ENK. And we hypothesized that ENK might be involved in the release of ATP. ENK might be doing something very similar to ABCC6. And all PXEers still have ENK. So that would be a good target to stimulate in PXEers if it would be involved in the release of ATP. So why did we think that ENK might be doing something similar as ABCC6? Well, that is because there are also a few patients that lack ENK. And we also have mice that lack ENK. And people that don't have ENK, they also suffer from pathological calcification or pathological mineralization. But it affects different tissues than the tissues that are affected in PXEers. Because in people that do not have ENK, the tissue that's in between the vertebrae of the spine, so that's in your back, that tissue mineralizes in these patients. And that results in a very richer rigid and stiff spine. Another problem that these patients have is that they have calcium phosphate precipitates in the joints of their hands and feet. And that's very painful. But that's again also ectopic calcification. It's also interesting to note that in your inner ear, you have bone-like structures. And in people without ENK, due to the ectopic calcifications, these bone-like structures in your inner ear, they also fuse. And therefore, these people become deaf. So to wrap up, ENK, people without ENK also have ectopic calcification, but different tissues, different organs are affected than the ones that are normally affected in PXEers. So we wanted to test, does ENK indeed, as we hypothesized, transport ATP out of the cell into the blood? And we did not immediately go to animal models. First, we studied cells that we cultured in a Petri dish. And what we did is we compared cells that did have ENK with cells that did not have ENK. And we could then collect samples of the extracellular milieu that these cells were in. And we could test if ATP was present outside of these cells. And that's shown in this panel. And what we found that is ENK was present, these cells released ATP into the extracellular environment. And similar to what happens after ATP was being released via ABCC6, also for the ATP released by ENK, the ATP was then converted into AMP and pyrophosphate. And this is the most important part of this story, obviously. So also ENK is involved in the regulation of extracellular pyrophosphate levels. So this was actually very encouraging data. And we then wanted to see, is this process also important in PXEers, or in basically in human beings, to prevent calcification in soft connective tissues? And to test that, we used mice that don't have ENK. So we have mice that don't have ABCC6, but we also have mice that don't have ENK. And what we did is we collected blood samples of the mice that don't have ENK, and we tested for the amount of pyrophosphate. We also collected skin samples of the mice that lacked ENK. And using a CT scanner, so that's just kind of sophisticated X-ray scanner, we also determined the amount of calcium phosphate that was present in pieces of skin of the animals that did not have ENK. If we first check what was happening in the blood of these animals, we found that animals that did not have ENK had reduced levels of pyrophosphate in their blood. Levels were not as low as those found in animals that didn't have ABCC6, but still it was a substantial reduction. And this was also found by another group that did the same thing. So that's the Budapest group. You will probably have heard of that. They also found that levels of pyrophosphate in blood of ENK lacking animals is lower than in control animals. So together, this indicates that most of the pyrophosphate that's present in the blood, so this important molecule that prevents ectopic calcification, is regulated by ENK and ABCC6. So, but does ENK also play an important role in prevention of ectopic calcification of soft connective tissues, so of skin, eyes, etc. So to determine that, we did micro-CT imaging, and we have a very, well, I would say a small micro-CT imager that's specifically designed to scan very small pieces of tissue, or for instance, mice. And what you can see here on these issues, this is the head of a living mouse. So this is, and you can see the white here, that's the skull of the mouse. And this is the mandible. And here you have the nose cone, because these animals are under anesthesia when we image them. And what we found is that animals that lack ink, they have a little bit of calcification in their skin, because this piece of the animal should not contain any mineralization. This is normal, because that's just the skull and this is the mandible. So that's all, that all should be there. But this little bit of calcification shouldn't be there. And there's more calcification in animals that didn't have ABCC6. And that also fits the fact that the levels of pyrophosphate in the blood of the ABCC6 lacking animals was more reduced than animals that only lacked ink. But look what happens in animals that didn't have either ABCC6 or ink. These animals really had dramatic mineralization in their skin. And we could quantify that, and we put that in a graph, and you can see that it's really a dramatic difference between the ABCC6 lacking animals and the animals that lack both ABCC6 and ink. It's maybe 40-fold more mineralization in these animals. And that was not only seen in mussel skin, but we also found that animals that lacked both ink and ABCC6 also suffered from extreme calcification in their ears. And that these studies identified ink as a pharmacological target in PXE patients. And what does that mean? It means that if we can find a way to stimulate ink in PXEers to release more ATP into the blood, then more pyrophosphate will be formed, and that should, in theory, then prevent the disease from progressing. So we now have updated our schematic overview how ABCC6 and ink participate in the regulation of pyrophosphate in the blood. And that is ABCC6 in the liver is involved in the release of ATP into the blood. In other organs, because ink is present in many different tissues, in many different organs, and there it can contribute to also release of ATP into the blood. And after release of ATP into the blood, you still need this other enzyme to produce pyrophosphate out of the released ATP. And the pyrophosphate then travels to the distant soft connective tissues to prevent calcium and phosphate to precipitate and to prevent ectopic calcification. Okay, so the final part of this webinar will be used by me to show some slides about how oral pyrophosphate might have a role in treatment of PXEers, or maybe not, because we are still working on that, and I'm not sure yet exactly if this will eventually work. And as said, we have shown in the past that there is some uptake in mice and humans, but most of it will be converted into inorganic phosphate. So inorganic phosphate might combine with calcium to form calcium phosphate precipitates in soft connective tissues. So I've had this hypothesis for quite some time, and now we decided to put this to the test. So what we did, we used our PXE mice, so our animals that lacked ABCC6, because it's an excellent animal model for human PXE, because these animals, they have calcification in their skin, they have calcification in their eyes, and they have calcification in their arteries. And what we did is we put pyrophosphate in the drinking water of these animals. And so all water that these animals took contained pyrophosphate. And the highest concentration of pyrophosphate that we tested was 90 millimolar. You don't have to remember these numbers, but the only thing that you should remember is that that's really a lot if you would translate this amount to a human PXE patient, because that would translate to someone of 70 kilograms. I don't know how much is that in pounds, 150 pounds. That would translate in 140 grams a day. And that's something like a couple of us. So that's really a lot of pyrophosphate. But after providing the PXE mice with pyrophosphate and treating them for half a year, so these were relatively long-lasting experiments, we then determined if the pyrophosphate prevented the ectopic calcification in the skin and in the eyes. And here you can see the results of this experiment. So the top shows the muzzle skin of an untreated PXE mouse. And the green material is soft tissue, whereas, whereas the blue material is the ectopic mineral that has deposited in the muzzle skin. So you can see that there's substantial calcification in the muzzle skin of untreated PXE mice. And that is depicted also for several animals. The amount of calcium phosphate deposition in the skin is shown here in this bar. So there's quite a lot of calcification in the muzzle skin. The PXE mice that received 90 millimolar of pyrophosphate in their drinking water showed no calcification in their muzzle skin, however, because we didn't find any calcification. Animals that received half of this dose, they were also pretty much protected against calcification. But if we went even lower, then we couldn't distinguish from animals that were not treated. And 45 millimolar, if you would translate that to human PXE patients, that is still too much to give to a PXEer. So then what was happening in the eyes of these animals? So here, and this is zoomed in, obviously, because mice don't have eyes that are this big. But again, the green is the soft tissue. So this is the eye. And this animal has been treated with 90 millimolar pyrophosphate. And there's no blue. So that means there's no calcium phosphate, no ectopic calcification in the mice treated with this extreme dose of pyrophosphate. The untreated PXE mouse, however, showed clear calcification, as demonstrated by these blue dots in their eyes. And we didn't scan all, this is an ongoing experiment, we didn't scan all eyes of the PXE mice yet. So we only have results of the animals treated with 90 millimolar pyrophosphate. But we could completely block the disease in the eyes. So that's positive. So some conclusions. If you give pyrophosphate to PXE mice, you need really high, at least in our hands, really high amounts to prevent calcium phosphates from precipitating. The good thing, however, the positive thing about our experiments is that they did not indicate that if you give a lot of pyrophosphate, that stimulates. That was my initial hypothesis, but that hypothesis was proven not to be true. So no increased calcium phosphate precipitating in animals that were given these extreme amounts of pyrophosphate. In the past, together with our colleagues in Budapest, we saw inhibition of the mineralization at lower amounts of pyrophosphate given to the PXE mice. And we don't have a good explanation why this is, but we have to look into that, because it might also give clues why certain treatments might work in one PXE patient and maybe not in another PXE patient. Also, the variability in calcification in the ABCC6 lacking animals was high, and that's also interesting because that might point to new ways that we can use to treat PXE. And something that I liked, because I have always been told that PXEers fear most what's happening in their eyes, that we for the first time show that calcification in the eye can be prevented. At least I think we are the first. And that's something I think is exciting. So what's still ongoing is we are currently testing other small molecules like pyrophosphate for their calcification inhibitory potential. And we have identified some that are way more potent than pyrophosphate. And they are also, let's say, natural compounds. So that's really exciting. We also are testing what happens if we give the PXE mice a dose of pyrophosphate once a day. We can just put that into the stomach of the animals, and that works actually it's, at least I should say, it's easy to give the animals the pyrophosphate using this procedure. And that's more reflects what would happen in PXEers if they would take a pill, because you're not going to walk with a water bottle full of pyrophosphate all day long, right? So you take one pill, at least that's what you hope, and then for the day, and that should help you. So we are now testing if that's possible. And together with the Budapest group, we will test or try to figure out why in different labs we get different results for the dose of pyrophosphate that we need to give to the PXE mice to prevent ectopic calcification of the soft connective tissues. Once more, what did I try to tell you today? Orally administered pyrophosphate inhibits skin and eye calcification, but extremely high doses are needed. And these doses are not realistic for human use. The protein ENK does something very similar as ABCC6. So we are now looking into ways to get ENK to work harder, to pump out more ATP, because that might also help PXEers. And as told earlier, other groups are trying different ways to get pyrophosphate up in the blood. For instance, a group in Boston tries to increase EMPP1 activity, but this will be an extremely expensive therapy, which is probably not affordable for most PXE patients, I fear. And there is a pharmaceutical company that tries to increase plasma pyrophosphate by inhibiting tissue non-specific alkaline phosphatase, so the enzyme that degrades pyrophosphate. So that was basically what I wanted to tell you today. And I want to thank a couple of people that contributed to the work that I presented today. And one very important one is Fateme Niasiorimi, my technician, who did a lot of the assays that underlie the experiments of which I showed you the results today. But also a couple of students who helped scanning the mice, so to pick up the calcification in the different tissues. Of course, Flora Tseri, who used to work with me here in Philadelphia. She's now in Hungary. And also late Joni Wito, who so sadly passed 1.5 years ago, because he really recruited me to join his group in Philadelphia and allowed me to set up my own group here at Jefferson. And that allowed me to continue my work on PXE and on other ectopic calcification disorders. So, and I finally want to thank PXE International for allowing me to share our work with the people that we're doing the work for, and also for their support over the years for our work. So yeah, I would be happy to take any questions, and I will do my best to answer them if there are any.
Great. That was wonderful, Koen. Thank you so much. Is there a long name for ink?
Yes, there is. It's progressive ankylosis.
Now that sounds like a medical condition.
The medical condition, there are few medical conditions associated with absence of ink. For some, there's not really a name because there are, let's say, only 10 patients known. It's only in one family. For others, it's craniometaphyseal dysplasia, CMD.
Okay.
And there are also not a lot of patients, but a little bit more than the family that I just talked about.
Okay.
So it's not, PXE is rare, but the conditions that are caused by absence of ink, they are even more rare.
Okay. Okay. And in the chat, Koen, we have a question from Helen. You mentioned blood vessels in legs, but do you see blood vessels elsewhere calcified, such as aortic arteries?
Yes, in the ABCC6 lacking animals, indeed, parts of the aorta can calcify. And this is in humans also reproduced by the group of Wilco Spiering in Utrecht, because we use micro CT to scan our mice, and he uses CT scanning to follow disease progression in human PXEers. And he indeed sees that many arteries can be affected in PXEers. Yeah.
Okay. All right. In PXE, is there an increase in TNAP and a decrease in ENPP1?
As far as I know, the reports are sometimes a little bit conflicting. I think there has been one study in which they reported increased TNAP activity, but that might have been only in cultured cells, I think. We have tested alkaline phosphatase activity, TNAP activity in blood of the different mouse strains that we have, and we do not see such a thing. So we don't see it. So we think the main problem is not increased TNAP, but TNAP is always there, because if you don't have TNAP, you would get too much of the pyrophosphate. And having too much is also not good. So completely blocking TNAP is not good. It's not a good idea. But that is not what this pharmaceutical company is after. You can easily inhibit, let's say, 75%. That should be no problem.
Okay. And did you see reduction in calcification in arteries with a daily dose of PPI?
So we have not scanned the arteries yet, because it's really very time consuming, because it's rather technical, but the micro CT scanner makes very detailed pictures of the organs that we scan. But the disadvantage of that is that you need to scan for a very long time. So it's very time consuming. So we have only scanned skin, and we started to scan eyes, but after that we will scan aortas and heart.
Great. All right. And what other substances inhibit calcification?
There are proteins that inhibit calcification. An example is fatuin A. It's a liver-produced protein, and that also is involved in prevention of ectopic calcification. Another protein that inhibits calcification is matrix GLA protein. That's a protein that's produced by various cell types. Other small molecules that inhibit calcification are, for instance, And some PXEers might have joined this clinical trial in which magnesium has been tested. Magnesium is a mineralization inhibitor. And we have identified some others, but I can for now not talk about these because we need to first file a patent about that.
Got it. Okay. All right. And another question. There has to be some difference in mechanisms other than PPI for ABCC6 and ENK because patients show calcification in different tissues.
Mm-hmm. That's right, but that not necessarily means that there are different factors, because it can be that in some patients, ENK is more active in certain tissues, and then in these PXEers, that tissue will not calcify, whereas in other PXEers, ENK is less active, and that might actually then explain why they show calcification in these areas. So that's not necessarily tells you that there must be other factors. It can be, I agree, it can be that ABCC6 does something else than being involved in release of ATP and pyrophosphate formation. Because, for instance, we have looked also into ENK, and ENK not only is involved in release of ATP, because ENK is also involved in the release of citrate. And we have published that. And so it can be that also ABCC6 is involved in the release of something else. We have looked in detail into this. We didn't find it, but the fact that we didn't find it doesn't mean that it's not there, this other substrate.
Okay. Is it possible for PXE patients to have no calcification showing in an MRI full body test of the arteries?
Yeah, so that's probably better asked someone that does these whole body CT scans.
Okay.
I think the answer is probably, it might be possible, at least if you consider people lacking ABCC6 suffering from PXE, because most often it's the diagnosis PXE is made based on the clinical manifestations, right? And then there are calcifications present. But I would assume, especially when people are still young, that it might be possible that some people don't have calcification or very low levels of calcification. Right. We even see that in our animals. I can go back, for instance, right? So here you see, these are all the same, because these are, you can see these PXE mice as all identical twins. So they're all identical. And still you see quite dramatic differences, you see, between the amount of calcium and phosphate that has precipitated in the skin. So that indicates that even with the same mutation, with the same genetics, there might be quite substantial differences in how the disease shows up.
Yeah. If magnesium inhibits calcification, would copious amounts of calcium, such as consuming large amounts of whole milk, also inhibit calcification?
No, no. Calcium does not inhibit calcification. No.
Okay.
No. And let's say, let me add to this that, for instance, taking a lot of phosphate or a lot of phosphate in mice, in the PXE mice, make the disease more severe. So that's also something to realize. Yeah.
Okay. All right. And okay, so that brings us to top of the hour. And I want to thank you and thank everybody for participating. And I think our next one is in October sometime. So, but of course, everybody will hear from me. All right. So thank you. Anybody else?
Thank you very much.
Thank you very much.
Yeah.
Welcome.
Thank you.
Yeah.